proceed with starting it. And it appears that we are officially live. So hello, everybody, and welcome back to another session of our Sussex Vision Seminar Series, as always, within the Worldwide Neuro Initiative. I'm George Kafetsis, a master's graduate from Thomas Euler's lab, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from Northwestern University, Professor Tiffany Schmidt. Tiffany's scientific path reflects her growing interest in retinal ganglion cells that sense light directly. Following her undergrad degree in biology and psychology, Tiffany joined Paolo Kofuji at the University of Minnesota for her PhD on the diversity of form and function of these intrinsically photosensitive retinal ganglion cells, the ipRGCs, as they are abbreviated. In 2010, she moved for some very successful postdoctoral years to Johns Hopkins University and the lab of Summer Hatter before joining Northwestern in 2014 and starting there her own lab. She has remained there since, and nowadays Tiffany holds the title of Associate Professor at the Department of Neurobiology and is the director of both the undergrad and the master's neuroscience programs. In her lab, they are interested in how light regulates behavior, and they tackle this question at different levels, from how cells function to where they project in higher brain areas, and to how or what actually they contribute to different visual behaviors. For such multi-level questions, they exploit the genetic accessibility of mouse models and employ different techniques from molecular to IFIs and behavioral to elucidate the role of these ipRGCs in both image and non-image forming vision. It is such a pleasure to have Tiffany here with us today, taking us through some of their latest and I'm sure exciting findings in her talk entitled Visual Circuits for Threat Anticipation. So without any further ado from my side, please all welcome Professor Schmidt. Tiffany, the stage is officially all yours.
Thank you very much, George, for the invitation to speak today. It's great to be back. Let me just get my screen sharing and everything organized here. Okay, so as George mentioned, our lab has really had a long-standing interest in understanding how light affects behavior, and we want to understand that question all the way from the retinas to the brain and kind of all of the circuits and cell types in between. So today I'm going to tell you about actually two ongoing projects in the lab, both from Marcos Aranda, who's an incredibly talented postdoctoral fellow in the lab, going on the job market in the next year or two. So watch out for him. And I'm excited to share this with you. So, light influences behavior through really two distinct systems. So we have image forming vision, which involves the detection and perception of color, contrast, form, and motion. And you have non-image forming vision, which involves those functions that occur outside of our conscious perception. So a really well-known one, non-image forming function is circadian photoentrainment, where organisms will align their daily activity rhythms to the environmental light-dark cycle in order to align an animal's activity and physiology rhythms to the environmental light-dark cycle. Another really important non-image forming function is the pupillary light reflex, which is really key for regulating the amount of eye that's entering our eyes and getting to our retinas across different luminances in the environment. And of course, in mammals, all of this information is getting to the visual system via the retina, which is this layered structure that sits at the back of the eye and consists of 6 different types of neurons. So as light comes into the eye, it's focused by the cornea and the lens onto the retina, passes through all of these layers of neurons, falling on the outer segments of these rod and cone photoreceptors, which will then convert that light information into an electrochemical signal that's relayed back through this network of interneurons to the retinal ganglion cells of the ganglion cell layer. And these ganglion cells project to 40 plus targets in the mouse brain, which I'm showing you a few of here. And these different structures in the brain are then sort of differentially regulating these image versus non-image forming behaviors. So we have areas of the brain involved in conscious visual perception or image forming vision, such as the dorsolateral geniculate nucleus, which is the visual thalamus, as well as the superior colliculus. And you have brain regions devoted to non-image forming functions. So the suprachiasmatic nucleus of the hypothalamus is one such region. So this is the master circadian pacemaker And this pacemaker is responsible for synchronizing all of the other clocks in the body, and there are many to this central pacemaker. And so light input from the retina to the SCN are what is then responsible for photo entraining your endogenous activity rhythms to that environmental light dark cycle. Another important non-image forming region is the shell of the olivary pretectal nucleus, which is important for driving pupil constriction in response to increases in environmental illumination. And so for a long time, this was kind of the way we thought things worked. You have light detection in the rod and cone photoreceptors, relay of that information by the ganglion cells to the brain, and then you have these image and non-image forming functions driven by these various brain regions receiving ganglion cell input. However, it started to become clear back in the 80s and 90s that this really might not be the whole story, that rods and cones may not be the only light sensitive cells in the retina. And these clues actually came from first patient data from clinician observations of blind patients who they noticed retained certain non-image forming functions, even though they were completely perceptually blind. So I'll show you one example of what that looks like here. This is work from Chuck Seisler's group at Harvard in 1995. And here you have a normally sighted subject and their plasma melatonin levels. And melatonin is the so-called sleep hormone. And so melatonin will be high during the night and low during the day in a normally sighted subject. And then if you give a person a brief light pulse in the middle of the night when their melatonin is high, you can see that their melatonin levels will sort of transiently and fairly rapidly decrease. This is one example of why it's really bad to give yourself a light pulse in the middle of the night as a side note, because this is what happens to your melatonin hormone levels. You can see that when the lights do turn off though, that level will rebound back up to sort of its normal level and then go back down when the day comes. So that's a normally sighted subject, but what they noticed was when they looked at these melatonin levels in blind patients who had degenerated rod and cone photoreceptors, so no known photoreceptors in the retina, those blind patients retain the ability to have high melatonin during the night, low melatonin during the day. And you'll notice that this blind individual also showed this transient decrease in melatonin levels to a light pulse in the middle of the night. So how do you have people lacking all known photoreceptors who are clearly still responding to environmental light cues, even though they're telling you they're completely perceptually blind? These people had no light perception. So it was these types of data that really clued people in that there was likely a third class of photoreceptor. Others recapitulated these findings in mice that lacked all of the rod and cone photoreceptors. And so then the search was on for really the identity of this new class of photosensitive cell. So taking a cue from the fact that the functions that were retained in these blind individuals were non-image forming functions, David Burson's group took a really simple but really elegant approach, I think, to trying to uncover the identity of these new photoreceptors. And what they did was retrogradely labeled the ganglion cells in the retina that projected to this central circadian pacemaker. They're doing this in rats. In this particular experiment, they put fluorescent beads in the SCN retrobeads, and this allows them then to identify the ganglion cells in the retina, which is shown here in B, that send input to this central pacemaker. And what you can then do is target those SCN projecting ganglion cells for electrophysiology and see if those cells themselves might be light sensitive. So when David's group recorded from these SCN projecting ganglion cells, they looked at the light response. These are current clamp recordings, and these steps up here are just when the lights turn on and then when the lights turn back off. You can see that these SCN projecting ganglion cells, if you look at the black traces first, depolarized in response to the onset of that light stimulus. They maintained their depolarization throughout the light stimulus, and actually long after the light stimulus turns off, these ganglion cells continued to fire. And this in and of itself is not earth-shattering. A ganglion cell should respond to light if you give it a light pulse and record from it. But what made these SCN projecting ganglion cells incredibly unique and special was that the Burson lab then recorded from these same cells, but in a cocktail of pharmacological blockers of all synaptic transmission in the retina. And that's what's shown in these top two traces in red. And you can see that even in the absence of any input from the rod and cone photoreceptors, thought to be the only photoreceptors in the retina, these SCN projecting ganglion cells continue to respond to light. And they do so with that same sustained depolarization that persists following stimulus offset. Indeed, even if they physically isolated the somata of these SEM projecting ganglion cells from the surrounding retinal tissue, you can see that the soma itself of this particular cell continues to depolarize in response to light. So these ganglion cells were truly intrinsically photosensitive and were given the very long name of intrinsically photosensitive retinal ganglion cells. So we call them ipRGCs for short. And the protein that allows these ipRGCs to respond directly to light is a photopigment called melanopsin. So like rod and cone opsins, melanopsin is a 7-transmembrane G protein coupled receptor opsin called OPN4. It is maximally sensitive to blue light, so at about 480 nanometers. And so ipRGCs are bona fide photoreceptors. They express a photopigment and they respond directly to light. And one of the things that makes them really fun to study in the retina is that they're also bona fide ganglion cells in the sense that they are getting input relayed from these rod and cone photoreceptors that they are then integrating with their intrinsic melanopsin response, which gives them some really interesting signaling properties that I'm not going to talk about today. And it's also important to note that these ipRGCs are actually incredibly well conserved. We see them, others have seen them using melanopsin immunolabeling in human and non-human primate retinas. And there's good evidence too that melanopsin is affecting the behavior of humans as well as mice and other animals. So if we look at the projections of ipRGCs and compare them to conventional ganglion cells, the ipRGCs are shown in this light red and conventional ganglion cells in gray, you can see that ipRGCs project quite widely throughout the brain. This is one of the reasons that I love studying this system, because ipRGCs project to so many different brain regions that I feel like by studying the ipRGC circuits, we can actually learn a lot about many different visual circuits. And we know from a lot of work from Sam or Hattar's lab, but also other labs that, and some of my own work, I guess, that ipRGCs are required for multiple types of behavior. So we know they're absolutely required to relay any light information for non-image forming functions. So if you ablate the ipRGCs, for example, animals will lose the ability to photoentrain, and they'll also lose the ability to constrict their pupils in response to light. So they relay the rod, the cone, and the melanopsin input to drive these non-image forming functions. But we also know that ipRGCs are involved in pattern vision. So they were originally discovered as sort of a circadian photoreceptor, but as we've learned more about the system, we now know that they're also involved in visual perception behaviors, along with the conventional ganglion cells. So if you ablate the ipRGCs or you knock out melanopsin, you get deficits in an animal's ability to, for example, see contrast as well, that they don't become completely blind, like the conventional ganglion cells play a very important role in visual perception as well. They're really influencing a broad range of behaviors. And so before I transition into the actual data, I just want to leave you with this thought that, we know about a few different behaviors that ipRGCs modulate, but really their broad projections throughout the brain suggest that there are many levels of modulation of ipRGCs on different behaviors that we just have not uncovered yet. And so that's what I'm going to talk to you about first today. And so we got interested in the question of how do animals decide what types of behaviors they actually want to take when they're acting in sort of their naturalistic environment where they're not always safe. So one of the interactions that is most fascinating, I think, in nature is this interaction between predator and prey. So predators have evolved multiple mechanisms for better detecting and capturing prey. And likewise, prey animals have evolved a lot of mechanisms for better evading or escaping from predators. And of course, the visual system is often central to this ability of these different prey animals to avoid predators. But I think equally important to the ability to sort of avoid detection or escape from a predator once you've been spotted is when you're making the decision as an organism about how much risk you want to take when you're entering a given environment. So you can imagine that animals are constantly faced with the decision of, do I enter this environment? And if I do so, I might get eaten by a predator, but I get to, obtain food or mate, or do I avoid this situation because it's too dangerous? But if that organism avoids that situation, it does so at the cost of getting food or of reproducing. So they're constantly doing this calculation. And of course, in order to survive, you have to be making the right types of calculations. So the system has to be really well calibrated, and animals have to be able to predict whether a threat is actually going to appear. So Markos got interested in this idea of how well an animal can actually predict the appearance of a threat. Thankfully, in the visual system, we actually know of a stimulus for mice that is innately threatening, meaning it doesn't require training for the animal to become afraid of it. And this is called the looming stimulus. So again, to mice, this is innately threatening, and it's actually quite simple to present to the mouse. So you can put them in this arena. Once they're in the center of the arena, you have a computer screen over the top, and you can play an expanding black disk over the mouse over multiple seconds, one expansion per second. And mice will find this innately threatening, and they will either freeze or they will escape. And it's thought to be innately threatening for the animal because it mimics the approach of a predator overhead, which would of course take the form of this big shadow expanding in the mouse's visual field. So Markos wondered if we could use the fact that this is an innately threatening stimulus to see how well an animal can predict the future reappearance of a threat in an environment that it's previously experienced. So this is the paradigm that Markos designed to measure something we're calling visual threat anticipation, or VITA for short. So what Markos does is he places the mouse in an arena for 4 minutes. The animal's never been in this arena before, so it's just a neutral context for the animal. He'll then play, after those 4 minutes have elapsed, one set of a looming stimulus exposure over 10 seconds, so 10 expanding disks of a single contrast over those 10 seconds. And then we just put the animal back in its home cage for 2 days. 2 days later, we come back and we just put the animal back in that arena, and it gets no further visual stimulation. And we see if the animal is acting like it's experienced, or that it's expecting the reappearance of this threatening stimulus that it experienced. So if it is anticipating the reappearance of that threat, the idea would be that that animal would avoid the threat zone where the threat had previously appeared, may also reduce the amount of locomotion, so reduce the exploration of the arena. So this is the anticipatory behavior that we're actually interested in measuring here during the test phase. So first, we wanted to measure the looming response of these animals in this paradigm in our hands, and we wanted to look at it to looming stimuli of contrast, from very low to 100%. So this is the percentage of animals that respond to a looming stimulus in our exposure phase, so this middle phase here. And you can see that as the contrast of the looming stimulus increase, the percentage of animals that are showing a detectable response to that stimulus in the form of freezing increases. And then you can also just measure freezing time, and you can see that as the contrast increases, the amount of time animals spend freezing also increases for that 10-second period. So we are able to generate a looming response, and it's important to note that this 55% contrast, which is very low, doesn't actually elicit a detectable looming response for the animals in our paradigm, and this will become important in a minute here. This is sort of a sub-threshold stimulus. Okay, so now what about the anticipatory response, this visual threat anticipation? What does this look like in a control animal? So if you put the animal back in that context 2 days after it's experienced one exposure to a looming stimulus of a given contrast, we chose this 55% contrast to start with. And very surprisingly, we found that even at this really low contrast, so fairly low salience stimulus where we couldn't even detect a looming response, we saw that animals would show anticipatory behavior. So on the left is an animal that received no looming stimulus, is just a 0% control. So you put it back in that arena 2 days later, and it still will freely explore the entire arena, compared to an animal that was exposed to 55% contrast looming stimulus. You can see spends much less time in the threat zone and a decreased distance traveled. So it's showing really strong anticipatory behavior, even to this really low contrast stimulus. And so the fact that this was happening at, this stimulus here at 55%, where we weren't even seeing a detectable looming response, clued us into the idea that perhaps this was occurring through a circuit that was actually separate from the looming detection circuit itself. So because we know that ipRGCs project widely to some of these non-image forming brain regions, we know that they signal over incredibly long time scales. This made us think that maybe ipRGCs were actually a candidate for sort of the visual inputs to modulate this anticipatory behavior over these sort of days-long time scales. And so we wanted to know whether ipRGCs are involved in this visual threat anticipation behavior. And so to start, if ipRGCs are involved in visual threat anticipation, then we should see deficits in this behavior when we use mutants where ipRGCs are dysfunctional. And so we started with a melanopsin knockout animal, which removes that really long-term light signal that you see in ipRGCs, their intrinsic light response. And we simply compared their behavior during the test phase of this paradigm. So first, I'm just showing you the control experiment where we looked at just exploration during the test phase of these control and melanopsin knockout animals. So that's 2 days after they've been exposed to, in this case, no looming stimulus. And both of them explore the arena at, they spend the equal amount of time in the threat zone and equal amounts of total distance traveled. But what about after they've actually been exposed to this low contrast looming stimulus? Remarkably, when animals lack intrinsic light sensitivity in their ipRGCs, they essentially lose the ability to anticipate the reappearance of this threat. So while a control animal will now avoid the center after it's experienced a looming stimulus and show significantly less distance traveled, you see that melanopsin knockout animals show increased time spent in the threat zone and increased total distance traveled, suggesting they have deficits in this anticipatory behavior, and that they are in fact required for this visual threat anticipation. And so we wanted to make sure that this wasn't actually just a visual deficit in an animal's ability to detect the looming stimulus. ipRGCs are not thought to be involved in the looming detection circuit, but we wanted to make sure that they could see the stimuli okay. And so we also looked at an animal's ability to just respond to the looming stimulus during the exposure to that looming stimulus itself. And we found that especially at these lower contrasts, which is where we're operating here for the behaviors I'm going to show you, you can see that the percentage of melanopsin knockout animals that are responding to the looming stimulus is similar, and the freezing time as the contrast increases between melanopsin knockout and their control litter mates was the same up until you get to these higher contrasts. So down in these lower contrast regions, we don't think that the changes or the differences between the two genotypes is a function of a decreased visual ability of some kind. We think it's a deficit in the actual anticipation of the threat, especially, again, down here at 55%. So one other thing I just want to touch on that I'm going to leave for a minute, and then I'll come back to at the end of the discussion of this part of the work, is that we saw some really interesting sex differences when we looked at this behavior. So initially, we had been grouping all of our male and female animals together, and that was where we first saw the deficit that I showed you. But Marcos then took and split his data by male and female mice, and remarkably, when he did that, he saw that it was actually just the male melanopsin knockout animals that showed that increased time spent in the threat zone, so that actually showed the deficit in anticipatory behavior. Whereas female melanopsin knockout mice had absolutely no difference compared to their control litter mates when we just split them by sex. And so we started off by pursuing, again, the circuit in the male animals, and then I'll circle back to the females. But this was a really good lesson for us in starting to split our data by sex. I would actually urge other people to consider doing the same, because we're finding the more we look, the more we find in terms of sex differences. And this was a pretty dramatic difference for us to see. And I'm glad that he thought to split the data and look at it in this way. So now we know ipRGCs are required for visual threat anticipation. We have this new behavior, and we wanted to figure out where in the brain are the ipRGCs actually sending this information that's tuning this anticipatory behavior. So we had no idea where to start, because we didn't even know what this circuit was. And so we wanted to do a screen to try to identify ipRGC targets that might be involved and which brain regions we should start to try to manipulate with different circuit tracing techniques. So to do this, we started off really simple with a CFOS-based screen. So we put our animals through our visual threat anticipation paradigm. They got either no looming stimulus or that low contrast 55% stimulus, put them back in their home cage for 2 days again, and then put them back in the arena for 4 minutes. like we would do their test phase. Then we just put them in the dark for 90 minutes, euthanized the animals, and immunolabeled their entire brain for CFOS. And then we looked at the different ipRGC targets throughout the brain with the idea that if a brain region is important for tuning visual threat anticipation, then the differences in CFOS activation should really mirror the differences that we see in behavior, where the 0% control and the 55% exposed melanopsin null animal will show similar CFOS, and then this exposed control animal would be different from the other two. And lucky for us, only one brain region showed this expected pattern of differences in CFOS labeling, and that was the perihabenular region. So here you're looking at CFOS density from immunolabeling, and you can see that the melanopsin knockout and the unexposed control have similar levels of CFOS, but this 55% control has significantly less than both. And so the perihabenula, if you're not familiar, is a region that was actually identified by David Burson and Samer Hattar's labs. It gets strong ipRGC input, and it kind of borders the lateral habenula, but is actually a distinct region from the lateral habenula itself. So now we have a place where we know ipRGCs are projecting that seems to be differentially activated during this anticipatory behavior across our different conditions. So we wondered whether these PHB, perihabenular projecting ipRGCs, are required for visual threat anticipation. If this is the case, then we should be able to rescue our melanopsin knockout deficits by activating the perihabenular projecting ipRGCs specifically, right? So we can specifically activate just those ipRGCs that go to the PHB, and that should rescue our melanopsin knockout deficit if these cells are actually required. So to do this, Marcos used a GQ excitatory dreads-based approach where he injected bilaterally into the perihabenula a Cre-dependent GQ dread virus into these melanopsin Cre animals, which are melanopsin knockouts. So by doing this, he's expressing the excitatory dreads only in the ipRGCs that go to the perihabenula. You can then activate the dread receptors on these cells using a ligand called CNO, which is specific for cells that are expressing the dread receptor. Because these are excitatory dreads, CNO will actually activate those cells. Okay. So excitingly, when Marcos specifically activated these PHB ipRGCs, animals went from having this more these deficits in threat anticipation where they're kind of crisscrossing the center to having significantly less time spent in the threat zone. Okay, so this is a rescue of the melanopsin knockout deficit by activating just these PHB ipRGCs. And he did this by first injecting CNO both when the animals were experiencing the looming stimulus, as well as when the animal was just put back in that context 2 days later. So then we started to wonder, well, when are ipRGCs required during this really long paradigm? Are they required when the animal's forming the association between the context and the stimulus, or are they required, or instead required, I should say, when the animal is put back in that context and needs to recall that that's where it experienced a threat? So to test this, Marcos did a really clever experiment where he just excited the dread receptors using CNO administration at either the exposure or the test phase to look at whether a single CNO application or injection could actually rescue the behavior. So we started with the exposure phase when the animal's actually experiencing this looming stimulus for the first time. And when he activated the GQ dreads during just this phase, he saw no difference between a vehicle injected littermate and a CNO injected littermate. However, when he injected CNO at only the test phase and vehicle during the exposure phase, that was when he was able to see a significant rescue of the anticipatory behavior, where the animal would spend less time in the threat zone, just like a control animal would, as long as we were exciting their PHB ipRGCs. This tells us that ipRGCs are required for, tuning visual threat anticipation appropriately, and the time that they're required during this behavioral assay is actually when they're having to recall that this is a context where I previously experienced this threatening stimulus, which I think is a really exciting nuance of this paradigm. So as a second test of whether this circuit was actually necessary for proper anticipatory behavior in visual threat anticipation, Marcos also did an experiment where he silenced the neurons in the perihabenula that get ipRGC input, which are primarily the GABAergic neurons of the perihabenula. And so in this case, he took the GAD2 Cre, this is a GABA synthesis enzyme, where GABAergic neurons in the PHB will express Cre, and then took a Cre-dependent inhibitory dread this time, and it bilaterally injected it into the perihabenula. So now instead of rescuing the behavior, he's going to silence the perihabenula. If the perihabenula is required for this behavior, then what we should see is increased time spent in the threat zone and increased distance traveled in animals where the PHB is silenced because they are no longer anticipating that threat. And that turns out to be exactly what he saw in the males. So you can see here the vehicle injected animals versus animals that had the inhibitory dreads activated by CNO. And so when the PHB neurons are inhibited, you can see increased time spent in the threat zone, increased distance traveled. These animals have deficits in their anticipatory behavior. So now we know the PHB itself is also necessary, which it should be if the PHB ipRGCs are involved. And so, yeah, and this is just to remind you that this deficit is mimicking what we see in the melanopsin knockout animals. So silencing that region mimics what we see when we take away the melanopsin from the ipRGCs. Oops. So now we know that ipRGC input to the GABAergic neurons of the perihabenula are really required for proper anticipatory behavior of these male animals. And so then we wondered what neurotransmitter ipRGCs might be releasing at the perihabenula to actually tune this behavior. In work I didn't introduce today, we have previously shown that ipRGCs are not only glutamatergic, which has been well established by many labs, but a subset of ipRGCs also releases GABA. And we know that they do this in the SCN and likely in the OPN to modulate photoentrainment and pupil constriction. But we haven't identified other behaviors that these cells are involved in as of yet. And a postdoc in the lab, Chen Liang, was able to use a mouse genetic reporter to show that these GABAergic ipRGCs do send a projection to this perihabenular region. So we wanted to see whether they were releasing glutamate or GABA, or maybe both, to actually impact this visual threat anticipation behavior. Lucky for us, there are conditional knockouts that allow us to remove either glutamatergic signaling by knocking out VGlut2 or by removing GABA synthesis by knocking out GAD2, specifically in the ipRGCs. So we have conditional knockouts that are OPM4 Cre VGlut2 flox or OPM4 Cre GAD2 flox, and these will remove the glutamatergic or the GABAergic signaling from ipRGCs respectively. So we simply started by just taking these animals and comparing their threat anticipation to that of their control litter mates. And really exciting, and this is again in males. And really excitingly, we saw that first of all, knockout of this vesicular glutamate transporter VGlut2 from ipRGCs caused a decreased anticipatory behavior in the form of increased time spent in the threat zone. So this mimics what we saw in the melanopsin null animals, right? The animals are spending more time in the center during that test phase 2 days later. Whereas these GAD2 conditional knockout animals actually showed enhanced anticipatory behavior. So they spent less time in the threat zone than did their control litter mates, suggesting that GABA signaling from ipRGCs is involved in tuning this behavior in kind of the opposite direction to glutamate release. Now we know that ipRGC, glutamate, and GABA are differentially tuning VITA behavior. We are at this moment completely assuming, I should say, that both of these signals are going through the perihabenula. But that's kind of our current working model that we would like to test more specifically. Okay, so now I want to circle back to the sex difference. So remember I told you that this sort of increased time spent in the threat zone, this deficit in anticipatory behavior was really only in the males when we split by sex, and the female melanopsin knockouts didn't show any difference compared to their control litter mates. But Marco started to wonder if maybe this is something that in females could be there, but maybe it's just something we're not seeing because we're not looking across the estrus cycle. So we did what was, I think, kind of a heroic experiment where for every single female he tested, he went through and retested a bunch of females, or tested a bunch of new females, I should say. He took vaginal swabs and looked and identified the stage of estrus of that female across every single day of the paradigm. So across all 4 days for every single female. And then he classified each female as either sexually receptive or not receptive on the actual day of the test itself. And when he did that, that's where we actually saw differences between the knockouts and the controls. So sexually receptive females, we saw no significant difference in terms of the time they spent in the threat zone, which is already really low. However, for these non-sexually receptive females, Marcos actually saw a significant decrease in the amount of time the animals spent in the threat zone. And if you're feeling a little confused, it's because for the males, the phenotype was the exact opposite. So for males, melanopsin knockout animals spend more time in the threat zone. For non-sexually receptive females, they actually spend significantly less. Not only is there a sex difference, but the effect is the complete opposite between these two different groups. Sex dependent and estrus dependent, just to make it like super complicated, but also very interesting. Okay, so is this effect in the females happening through the same circuitry as the males, or is it something kind of completely different? So we started with some of the parallel experiments to the ones I showed you in males. And the first was to try to rescue the behavioral deficits we see in females. Oh, that's the opposite graph. I should ignore that graph. That's like the male graph, not the female graph. So if this is important for females, then activating these PHB ipRGCs with GQ dreads could rescue the anticipatory behavior by actually increasing it. And so that turns out to be exactly what Marcos saw. So if you activate these PHB ipRGCs in females, they go from avoiding the center of the arena during the test phase to now kind of crisscrossing it a little bit more than they were, right? So you have this significant increase like we saw for the female, or to rescue the female deficits in threat anticipation. So this means that the same ipRGC PHB circuit is functioning in males and females to differentially drive this anticipatory behavior. So this was just a reminder of what we saw in males. These are the actual correct graphs for males here. So you have the rescue for males that resulted in sort of a decrease in time spent in the threat zone back down to control levels, rescuing this melanopsin knockout deficit that we saw in the males. And I should also mention that we see the same effect when we silence the perihabenula, where the females and the males go in the opposite direction. So what we've been able to identify is a new circuit where ipRGCs are involved in anticipatory behavior, where they're anticipating the reappearance of a threatening stimulus where in an environment that they previously experienced that threat before. And we're seeing that in males and females, the ipRGCs seem to play different roles in tuning that threat anticipation. And in the males, you have glutamate and GABA released, presumably in the perihabenular region, to differentially tune this behavior. So we're really excited to figure out what's downstream of the perihabenula. Marcos is doing a lot of work to figure that out, to specifically look at release in the perihabenula and even record in vivo activity to see what's actually happening when the animal's behaving at these different phases. So now I just want to switch gears and tell you briefly about a second project, actually also from Marcos, where we're looking at ipRGC diversity. So I've had since the beginning, I feel like I've been interested in ipRGC diversity and sort of what are the properties of these cells, what are their roles in behavior. And I've always been interested in what actually drives that diversity developmentally, what makes these cells diverse. And then just generally for neurons, what are the things that drive the development of subtype defining features for different neurons. So we got interested in this question for ipRGCs, right? So we know that they express melanopsin, but we also know that there are at least 6 subtypes of ipRGCs that have been identified. These cells differ across their physiological features. In almost every measurement you can make, there'll be a difference across the subtypes. They differ in their melanopsin expression level. So M1 cells have the highest melanopsin expression levels, then M3, then M2, and then M4, 5, and 6 have the lowest levels of melanopsin expression. They differ in their morphology. So M1 cells are the simplest and have some of the smallest dendritic arbors, whereas M4 cells have the largest and most complex dendritic arbors. They differ in their stratification. M1 cells are off stratifying, M2s and 4s and 5s are on, and 3s and 6s are bistratified here. So lots of different cells with lots of different properties. And what I like about using ipRGCs as a system for understanding cell diversity is you have represented here a lot of the different features that you'd find in a given ganglion cell. And then you have the fact that ipRGCs project so widely throughout the brain. They're driving a diverse array of behaviors that we can measure really nicely. So we're really poised to figure out a lot of things about ipRGC development if we can start to get at what the underlying genetic programs might be. We also know that ipRGCs are transcriptionally diverse. So work from Nick Tran and Josh Sains's lab has shown that multiple different ipRGC clusters fall out when you do 10x single cell RNA sequencing experiments. Our collaborator Yue Yang has performed a dimensionality reduction, and then we're showing here just the ipRGC clusters kind of remapped, and I've moved the M5 from in their paper where they put it to, we think it's down here based on some of Greg Schwartz's data on the Pixon cells. So we have the M2, the M1, the M2, and the M4 clusters here, and then we're kind of provisionally labeling these clusters, but we don't have any verification of that yet. So they're also not only morphologically and physiologically distinct in their projections, but also in their sort of genetic programs and the genes that they're expressing. So how do these diverse ipRGC properties actually develop? Well, one thing that interested me right from the start was the fact that Brain3B, which is a transcription factor important for ganglion cell specification, actually distinguishes different subsets of ipRGCs, and it does so in the adult. So we know that Brain3B positive ipRGCs include all of the non-M1 ipRGCs, which are thought to be involved in pattern vision. They all express Brain3B, as do a subset of the M1 ipRGCs that are absolutely required for pupil constriction. And then you have a small subset of about 200 M1 cells that are Brain3B negative that we know are sufficient to drive circadian photoentrainment. We have this really early expressed transcription factor that's for some reason being maintained into adulthood in a large proportion of ipRGCs. And so we wondered whether Brain3B expression levels might correlate with some of these features that really define the different ipRGC subtypes. And so this work was started by Stolke Lee, and then has been taken over since by Marcos. And she started looking at whether these ipRGC properties actually correlate with Brain3B expression levels. So here's what she did first was just label for the Brain3B protein using an antibody stain. And she found that, Brain3B fluorescence intensity is really not a binary thing. So we often present it when we talk about the mouse genetic models as like they have Brain3B or they don't. And I guess that's true, but the ones that have it have a pretty broad range of amounts of Brain3B protein. You can see generally the M1 cells have less than the non-M1 cells, which are just plotted here in pink. But really interestingly, when, and this is just like a Brain3B negative cell, a weakly expressing cell, and a highly positive M1 ipRGC. But Stolke actually filled each of these cells and looked at their morphological properties as well. And she found that among the M1 ipRGCs that she filled, there was actually a correlation between the amount of Brain3B and the size and the complexity of the dendritic arbor just within this one subtype. So we can quantify that here. So she's looking just at the correlation for the M1 cells. We just plotted the non-M1s for kind of reference. But you can see dendritic length and dendritic field both significantly correlate with the amount of fluorescence intensity within the M1 cells themselves. So this was a clue for us that Brain3B really might be driving some of at least these morphological features for sure within the M1 cells, and then maybe across some of these other ipRGC subtypes as well. Okay, so now we have Brain3B expression having this nice positive correlation with dendritic arbor size and complexity. And we also, she also observed, but I'm not showing you the data here, that the Brain3B negative M1s had the strongest levels of melanopsin expression, and actually the highly Brain3B positive M1 cells had lower melanopsin expression. So she's observing this interesting inverse correlation between melanopsin and Brain3B as well. Okay, so now we know this is happening, in a correlational manner with multiple features of the M1 ipRGCs. But what about the other ipRGC subtypes? Okay, so we know that these are all Brain3B positive. We're just like vague about how much Brain3B, right? And so do we see these same relationships? Well, first, what Yue did was just look at the levels of melanopsin expression using the SANES data set versus Brain3B expression, and you can actually see this really nice inverse correlation between the different clusters of melanopsin and Brain3B. That's, so they're nicely inversely correlated. And actually, if you look at just the properties that we already know of about M3, M2, and M4 ipRGCs, we actually know that the dendritic arbor complexity increases, the degree of synaptic input to these cells increases, and the melanopsin levels decrease across these different subtypes. And we know that their Brain3B levels are increasing from M1, M3, M2, and M4. So some really interesting relationships started popping out across a lot of different features that got us excited about the idea that Brain3B might be a really important regulator of the development of ipRGC subtype features. So this is when Marcos comes in, and what we really wanted to do was test this in a causal way. Instead of just identifying these relationships, is Brain3B actually causally affecting these different properties? And we did this work too in collaboration with a neighboring lab, Dr. Yue Yang, and then his student Omar, and a staff scientist in the lab, Tomoko, and actually another student just joined on, Shinichiro, who I don't have pictured here. And so we wanted to know whether altering Brain3B expression in the ipRGCs will influence the development of their properties. And we kind of started really broad with the idea that like it could be physiological, morphological, melanopsin, genetic programs. All of these things we know differ, so we want to test them all. And what we decided to do to test them was to either remove Brain3B completely from all the ipRGCs using a conditional knockout approach with a mouse line from Tudor Badia's lab, or we generated a new mouse line that would overexpress Brain3B in all ipRGCs by expressing Brain3B in a Cree-dependent manner at the ROSA locus. So now we can dial Brain3B all the way up or all the way down in ipRGCs, and we can see if we see a change in the features. And the really simplistic idea we had from this was that if we take away Brain3B, the features and the genetic programs and the morphology of ipRGCs would shift toward the M1 cells. And if we dialed up Brain3B, the features, the genetic programs, and the physiology would all shift toward, more toward the M4 cell population. And so, like I said, this is really ongoing work, and I'll show you some of the data that we have so far. So first, we wanted to look at melanopsin expression in these Brain3B conditional knockout and conditional overexpressing lines. And so we used RNA-scope fluorescent in situ hybridization to look at mRNA levels for melanopsin and Brain3B in individual ipRGCs. And first here on the left is Brain3B, and you can see in the conditional knockout, we have almost no Brain3B per ipRGC, which is good. That means our knockout's working. And you can see a significant increase in Brain3B in our overexpressing line. And then you can see that consistent with our hypothesis, melanopsin goes in the opposite direction of Brain3B, right? So if we knock out Brain3B, we see a significant increase in melanopsin mRNA in individual ipRGCs. And if we overexpress Brain3B, that suppresses the melanopsin expression levels, and it's significantly less than it is in control. It's not like melanopsin goes all the way to the ceiling or all the way to the floor, but it shows us that Brain3B is tuning the level of melanopsin, which is a really important subtype defining feature of the various ipRGC subtypes. So we've also looked at protein expression to see if this is mirrored in antibody staining of individual ipRGCs again, and you can see that when Brain3B is removed, we have an increase in melanopsin, which is shown here in the middle panel. And when Brain3B is overexpressed, we have a suppression of the levels of melanopsin in individual ipRGCs, which is easy to appreciate from this image here on the right. It seems that Brain3B is an important modulator of melanopsin expression levels in ipRGCs. What's actually really cool, but I didn't have time to put it in here, is that even if we remove the Brain3B or overexpress it in adults, so we can do this at P60, we actually see similar changes in the melanopsin expression levels. So it's not just a developmental thing, but it's something that's being sort of actively maintained by Brain3B in some way in the individual ipRGCs. So this is a pretty targeted approach looking at the melanopsin expression levels, just because we know it's a gene whose expression differs across the subtypes. We wanted to take a more unbiased approach and just look at the genetic programs of ipRGCs more broadly and see how those might change when we knock out Brain3B. So these are just the replotted UMAPs of the subtypes, and you can see if we just plot melanopsin expression, again, this is from the SANES data set, which has just been like the most useful thing for like our research lately, and it's such a gift to the field, I think, in general. But you can see the melanopsin levels and the Brain3B levels, the inverse correlation is really evident when you look across the ipRGC clusters. And then what's plotted down here in the cyan are sort of the top genes expressed in the low Brain3B ipRGCs. So M1 and M2 clusters, you can see, are more enriched for these genes. And then here, you guys just plotted the top Brain3B high defining genes here in this pink color. Just that'll be important later. And then we decided to compare the genes that were being actively translated in ipRGCs in control animals and animals where brain3B had been removed. So we used a trap-seq approach, which I will not go into the details of, but if you're interested, I can talk about after the fact, to just look at bulk ipRGC gene expression in our conditional knockout and our control animals. And there's that. And then what we see is that, wait, where did my graph go? Oh, thank goodness. What we see is that when we take away brain3B, the gene expression programs that are enriched in these brain3B high cells decreases in just the whole ipRGC population. So the cells overall are becoming less M4-like when we take away brain3B. And at the same time, ipRGCs in these brain3B conditional knockouts are also turning up genes that define those brain3B low ipRGCs. So in other words, they're not only becoming less M4-like, but they're also becoming more M1-like. So gene expression programs are indeed shifting more towards that of the M1, the M2 cells, rather than the M4 cells in the absence of brain3B. So what this is telling us is that brain3B is not just affecting melanopsin expression, but it's actually potentially a sort of a master regulator of the molecular identity, the genetic identity of these different ipRGC subtypes. So we're going to do follow-up experiments in the future with NIH's blessing, hopefully someday here, to look at how this is changing in a really nuanced way and try to understand what the actual genetic programs are that are underlying this shift, more at a single cell level. So that's sort of the genetic expression in these cells, but does removal of brain3B actually shift some of the morphological and physiological properties of ipRGCs? So we've started with morphology for now, and we've done this for brain3B conditional knockout animals. Again, the idea is you take away brain3B and everything's going to shift from the sort of large, complex dendritic arbors to the simpler, smaller dendritic arbors to become more M1-like. And so Marcos has been able to do this using sparse viral labeling of M4 and M2 ipRGCs, and we can, of course, distinguish these two based on whether they express this marker SMI32. So M4s are SMI32 positive and M2s are SMI32 negative. Both are on-stratifying. And so we compared M4 and M2 cells. We kept the region of the retina constant in control and brain3B conditional knockout animals. And really excitingly, the M4 cells in the brain3B conditional knockout and the M2 cells, you can see, both showed smaller and simpler dendritic arbors compared to their control counterparts. So here's a more expanded analysis for the M4 cells. You can see the shoal analysis showing decreased dendritic arbor complexity, significantly smaller dendritic field diameter. And then we have trends, this is still a data set we're collecting toward smaller total dendritic length and even smaller soma diameter when we take away brain3B. And M2 cells likewise show a significant decrease in dendritic field diameter. So this also seems to be important for the morphological development of these cells. And so what I've shown you so far is that brain3B is really shaping, some of these subtype defining features of ipRGCs. We have brain3B levels varying in a predictable gradient with ipRGC morphological and functional properties. They're inversely correlating with melanopsin expression levels. And then when we manipulate brain3B expression, which I should mention is happening post-mitotically, we've confirmed that in experiments I didn't have time to show you. This alters the melanopsin expression in ipRGCs. It's important for the molecular identity in shaping the molecular identity of ipRGC subtypes. And it's influencing dendritic development and even soma size, which I didn't have time to include today. And so lastly, in the last 3 slides, I just want to talk about the projection patterns and behavior as well, because we have really differential innervation of various brain regions based on whether ipRGCs are brain3B positive or brain3B negative, and of these different subtypes. So what Marcos did was use a Cre-dependent virus to label ipRGCs so that we can trace their axons to the brain. And then he looked at the contralateral and ipsilateral OPN and also DLGN, and then looked at pupil constriction as well as contrast sensitivity. So this is the control animal. You can see the contralateral. You have the core is really nicely filled here of the OPN, and then ipsilateral, you can see the shell. And when brain3B is removed, here you can see that you have a kind of a disorganization of the contralateral side, but I think what's really interesting is this ipsilateral projection, where you can see you've essentially lost the shell of the OPN projection. And now it's almost like you have another projection to the core on the ipsilateral side when brain3B is removed. And so Marcos has done some pupil constriction experiments as well. And so if you look with increasing light intensity at the pupil area, which of course gets smaller as the pupil will constrict, you can see at bright light intensities, the animals show a significant deficit in pupil constriction, where they don't constrict their pupils as much as littermate controls. So it's not a massive deficit, but there's definitely still a projection to the OPN, but there is a significant decrease in pupil constriction. So what about an image forming brain region? So Marcos has also mapped the projections to the visual thalamus, the dorsolateral geniculate nucleus. You can also see here IGL and VLGN. This is the ipsilateral projection, and then of course, or the contralateral projection, and then you of course have this ipsilateral projection here as well. And so interestingly, in brain3B conditional knockouts, and we know that these brain3B positive cells go to the DLGN, you can see a marked reduction in the projections to the dorsolateral geniculate nucleus on the contralateral side here. So you have decreases in projections to this image forming brain region. We haven't been able to do a test of conscious visual perception yet, but we have done a test of reflexive vision, which in our experience in other models, they tend to mirror each other in their deficits. And we've looked at contrast sensitivity using this optokinetic tracking paradigm from Glenn Prusky's lab, the optometry system. And in that assay, we see a significant decrease in the contrast sensitivity of brain3B conditional knockout animals where ipRGCs lack brain3B compared to their control littermates. So we're going to follow up on this and do some tests of conscious visual perception as well. But based on this, I would expect to see deficits, especially based on the anatomy that we're seeing with these animals. So it seems that ipRGCs, or that the brain3B in the ipRGCs is also really important for their projections and their behaviors as well, based on these initial experiments anyway. And so with that, I want to stop and take any questions. And first want to just thank the people that actually did the work. So here's Marcos, super talented, 2 super interesting projects, and it's been just so much fun and so exciting working with him on both of these. This is Lucy Liu, one of the undergrads who helped with the project, Chen, who showed the projections to the perihabenula, as well as the rest of the lab, and our collaborators here. And of course, I want to thank our funding as well. So that'll take questions. Thank you.
Thank you very much, Tiffany, for this amazing work, both in quantity and in quality. And working with non-model species myself, I cannot help but be very jealous of the resolution you have when it comes to manipulating things and checking what is the effect of this and that. So let me go ahead and post the Zoom room link for the audience in case they would like to start joining us already. And as a reminder, after the first round of moderation, of online moderation of the questions that appear, we will be continuing in an offline fashion in the traditional get-together informal Zoom gathering that we have. So waiting for questions to appear. The first one that I have, with respect to the second part of your talk, because you said that it's not a developmental stage-specific effect, this dynamic balance between the two, and it also happens postnatally. Would you expect? What would you expect the role of such active dynamic balance between the two would be in terms of melanopsin and BR, the factor, the other?
What the role of that will be?
Yeah, to actively be.
Oh, like the ongoing. Yeah, that's a good question. I don't know if it's if it's helpful to the cell to have that dynamic balance, or if it just is, and maybe that's what kind of gives it sort of the fine tuning. I would love to know if how important is the level of melanopsin expression for, say, an M4 cell? If we overexpress melanopsin in that cell, I wouldn't do it using the brain3B model, but if you just specifically overexpress melanopsin, is it worse? Is the contrast sensitivity of the cell or the animal actually affected? You would predict if it needs to be this well regulated, then it probably matters a lot for the function, but we haven't tested that yet. But it's a really interesting question.
Yeah, because I think what we know from classic textbook development is that the paths kind of get into one trajectory and that's it. they cannot actively maintain a balance. So yeah, it's very interesting. And then going to
it's a cool example of like, I think recycling, right? Like, because you have this pathway that was important to turn things into a ganglion cell, right? And that was kind of what Brain2B was thought to do. And then it's like, all right, now I'm going to use it for all these other things and just keep it turned on into, you know, adulthood. And I just love the repurposing.
Definitely, yeah. Going back to the first part of your talk about threat anticipation, I was wondering, and maybe like my naive understanding of how CFOS protocols work will be making my question very stupid. But if I understood correctly, like you managed to identify the perihabenular region based on the CFOS activity, right? But if GABA was the primary driver of such behaviors, the fact that CFOS only captures the activation, would it make would you miss some territory, some brain region, if GABA was behind it?
Yeah, I think we're very lucky that worked. Actually, I couldn't believe that it did. I mean, it was handy that we saw the patterns, right? But then it was like, well, unless we see a functional confirmation of this, like I wouldn't just trust the CFOS. And everything we've done with the genetic manipulations mimics sort of the VGlut2 knockout and the melanopsin knockout. And so that's the thing we don't quite know yet, is whether the GABA signaling is happening at the PHB, and we have to do some fancy tricks to try to just knock it out in those PHB ipRGCs if we want to get at that question. So we could be missing something. The things we did after that make me comfortable that it's like a major region. But yeah, for the GABA release, I agree, and it would be hard to see the CFOS if it's inhibiting. But then theoretically, the other areas would have had the opposite effect. So we might have still been able to pick it out just based on the comparisons with the different genotypes.
Yeah, I see what you mean. Thank you. So the first question that appeared in the chat is from David Berson, who uses a very nice interplexiform name for his YouTube account. So he says, fabulous talk. If I remember right, there should be many other types of BRN3B expressing RGCs, which are non-ipRGCs. Do you think the gene plays the same role there?
I would love to know the answer to that question. And I think I think one thing I would love to do in the future, I just want to make sure we do it in an intelligent way in terms of which cell type we would pick, and we would need a driver to do it, would be to, overexpress or knock out Brain2B in some other types of ganglion cells, and just see if we can drive, expansion of the arbor complexity by overexpressing Brain2B or something like that. It may or may not work. It's hard to, without genetic drivers, it's harder to do depending on which cell type it is. But yeah, it could be, right? It could be if it's kept on. I would imagine it would do some of the similar things.
Ping-ponging back to the first part of your talk. So my question is because this looming stimulus has a certain speed, right? Would you expect a tuned curve of speed preference for these cells that drive this behavior? So if you try different speeds.
I don't know if Marcos tried different speeds. This took us about a year to convince ourselves this was a real thing and make sure that the paradigm, was repeatable and clean. And so we haven't done a curve of that like we have for the contrast, because every animal we can only do one set of contrast. And so we've generated kind of a full curve at this point for males and females at both stages of estrus, actually. And so that's been huge, a huge data set to collect. But I think there's probably a lot we could tweak to try to figure out, is there any difference in the preference or the strength of the inhibition, or of the anticipation.
Right. Thank you very much for addressing this. I have one last question, and if there are no more questions appearing in the chat, given that people are already waiting in the Zoom room, we could terminate the live broadcast and continue offline. So if no more questions appear after my last question, I will be doing that. So my last question is because you quantify in terms of behavior, how many times, not how many times, how much time the animal would spend in the zone. Have you tried to normalize it by how many times it enters? Because it could be entering, walking around and spending some time, or staying even stationary inside, but this could be happening once, while other animals might not, might do it 10 times, compared to control versus that.
That is definitely, so Marcos has actually just popped into the Zoom room, so we can talk to him more about this offline. He's definitely done a much more comprehensive analysis, and we're gonna go back and do some of this more nuanced analysis on the behavior, just to try to see what other parameters might be important. I know he did a principal component analysis to try to pick out sort of the top behaviors, and those two were two of the main ones that we actually measured that popped out as principal components, and those are the ones I presented here. But that would be one that I could imagine maybe being different as well.
Right. Okay. Thank you very much for addressing this. Thank you very much for this fantastic talk again. And I'm so happy that Marcos is here so we can continue offline. And again, I would like to remind to our audience that if they want to be part of this offline discussion, they should make sure they click on the link that is available. Thank you very much.